Good evening, everybody. Welcome to another Darwin College lecture. Our speaker tonight is Keolu Fox, an assistant professor at the University of California, San Diego, and he's affiliated not just with the Department of Anthropology, but also the Global Health Program, the Climate Action Lab, and is the co-founder and co-director of the Indigenous Futures Lab. He's got a PhD in genomics from the University of Washington, and in Seattle, that is, and his multidisciplinary research interests include genomic sequencing, genomic engineering, computational biology, evolutionary genetics, paleogenetics, and indigenizing biomedical research. So he's published a great deal, as you can imagine, on human genetics, biomedicine, ancient genomics, and indigenous data sovereignty. We are delighted to have you with us tonight. So it's a great pleasure to welcome you onto the stage to give your lecture, which you are giving in honor of the legacy of Charles Darwin at this university. And your title is Wayfinding Through the Human Genome. Keolu, please come.
Okay, can you guys hear me well? Amazing. First and foremost, I'd just like to thank everyone for having me here. It's a true pleasure. I came from California. I'm originally from Hawaii, and I'm a huge student of natural selection and Charles Darwin. And to stay in the family's home and join you here in Cambridge is just a dream. So first, from my culture, you never go anywhere without giving people a gift. So I have, for Janet, I don't know where she is, but we have some of Mexico's finest tequila here for you. Thank you so much for organizing my accommodations, my, my room and board, figuring out dates, hounding me on email, and it's the good stuff. So, yeah. Then, for our two conference organizers, I have for you, very special, one for Alan. Here you go. Stephanie, here you go. Okay, so this is called codes, and we work on the ultimate code, so it's kind of cheating because we work in genomics and genetics, but those are not just lays. So usually, I would give you a flower lei that smells really good, but it's sort of ephemeral. It, only lasts so long. Maybe that's by design. Maybe that's Indigenous Futurism. Maybe that's how we should be thinking about design on this planet. However, those are seeds from Nukuhiva in the Marquesas. So for every 9 people who visit Antarctica, 1 person makes it to the Marquesas. And they're seeds, and seeds represent our future, and they represent the genetic code, and I think they're probably and easily one of the most elegant things ever designed by natural selection and evolution. So thank you for having me. All right. I'm from Kohala. Has anyone ever heard of Kohala before? It's on the Big Island. It's the northernmost tip. It's a really beautiful place. We have an interesting history there. One of the things that you should know about the Big Island of Hawaii is, one, from the top of Mauna Kea to the bottom of the ocean is the largest geographical feature on planet Earth. That is where Captain Cook passed away, but we'll get there. It's also one of the epicenters of biodiversity and biocomplexity on planet Earth. With 11 out of 13 biomes on our planet, you can snowboard on the top of Mauna Kea and surf without a wetsuit in the same day. We have high elevation. We have some of the most diverse soil profiles in the world. White sand beaches, black sand beaches, green sand beaches, you name it. They're stunning and incredible, and if you do have the chance to visit, drop me a line. But it's also an extremely complex place because it happens to be the simultaneous extinction capital of the world and the invasive species capital of the world. We have been dealing with other people's waste for a very long time. We have been dealing with the impact and the simultaneous impact of the military-industrial complex and the tourism-industrial complex. We have all types of problems, and it's impossible for young people and young scientists to grow up and be nurtured in that place and not make those types of observations. That's what's shaped my thought process. And it's also the most diverse place in America. We have the highest percentage of mixed race or mixed ancestry people in America by 25%. We have the county I'm from, District 1, Hawaii County, is the most diverse county in America according to our new census data. Followed by Maui County, which is number 2, Kauai number 4, and Honolulu at number 11. And here's this like beautiful tapestry of all of these different districts of people and the different communities that have come together. First, our Polynesian ancestors arrived from Tahiti, Marquesas, and the far-flung island archipelagos of the Pacific. Then we have waves of people coming from the Iberian Peninsula, Madeira, Azores, the English. We have people arriving from America, Asia, Okinawa, Korea. You get the picture, right? And so it happens to be this incredible place to understand really complex questions in human genomics. We have to design specific algorithms. We have to have an attention to detail in terms of allowing, our history and what we know of history to inform how we analyze data long term. Okay, so that's where I'm coming from. Currently, I work at the University of California, San Diego. You guys are currently ranked number 1 by World News. Cambridge University is the number 1 university in the world. We are ranked number 1 for the last 10 years by Surfer magazine. And a lot of modern genomics technology has its origins here, right? Watson and Crick, Rosalind Franklin, Frederick Sanger, his 2 Nobels. I could go on and on and on. Richard Durbin, all of these people who have directly impacted the development of the field. But we said in La Jolla that we'll take it from here. So we have the largest service provider for genome sequencing in the world, Illumina. We have, whether you like him or not, Craig Venter. We have a lot of new directions for technology development. And every year in America, we have the number 1 ranked oceanography institute, Scripps Institute for Oceanography. A lot of the things we learn about climate change come directly from this institute. Walter Munk, his D-Day calculations, the Keeling curve, Roger Revelle. So the types of thought and the way we approach climate change and genomics is like pretty natural, And our campus is built on Kumeyaay Nation, which is an indigenous community of people. And for that reason, we started the Indigenous Futurism Institute. So a lot of today's talk will be about how geography has shaped our genomes, specifically about geographically sequestered communities, those of indigenous ancestry. But we also focus on how we're shaping the environment. So here's a picture that I think is very interesting, and it shows Yosemite National Park in 1872 and 2020. What do you notice? Anyone want to point anything out? It's fairly obvious. Anyone? More trees, because they were maintained by indigenous communities that had access to that land. Because of that, they were able to nurture the soil. They were able to ensure and implement cultural burns. And they were able to clean up detritus and maintain the sort of harmony and balance of the ecosystem. On the right, that's what happens when you separate indigenous people from biodiversity and land. In fact, indigenous people who represent maybe 5% of the world's population are responsible for maintaining 80% of its biodiversity. And this is just an example of what happens when we neglect our ecosystems. Because as we say in Hawaii, the land is the chief and we're its servant, not the other way around. We don't need to Yeah, you get the picture. But for the full intents and purposes of today's talk today, and my little love letter to Charles Darwin, is that today, now, using a lot of the insights that were gained from his trips on the Beagle and his writings and many of the things that he's categorized and popularized in terms of thought, we've been able to identify the global distribution of many different locally adaptive traits. So I'll give you some examples. EPAS1 is a gene that's under selection in Tibetan and Nepalese people. It's a high elevation adaptation gene. It allows people to subsist in the Himalayas in ways that people like George Mallory would never be able to understand, because their genetic hardware is different and they, metabolize oxygen differently. CREBRF, found in the Pacific down here in Samoa, you can see, it is predictive of increased obesity, but protective against type 2 diabetes. It is a gift from our voyaging achievements and accomplishments. Sickle cell anemia and HBB, that is a classic example of the impact of a pathogen like malaria and the reduction of erythrocyte surface area leading to anemia, but also protection against malaria. So you get the picture. At this point in 2025, by sequencing new populations of people, we are identifying incredible new biomechanisms that allow us to understand not just our diaspora, migratory history, and deep past, but also our susceptibility to disease today. So I want you guys to focus on that, and that's very, very important. And there are many other examples, and this is from one of my professors from undergrad, Sarah Tishkoff. She's at the University of Pennsylvania now. But these results are in stark contrast of what I've just shown you. So take a look at this little chart. You can see that of the total participation in genome-wide association studies amongst human beings, according to Melinda Mills' GWAS Diversity Monitor, 95% of these studies exclusively feature individuals of Western European ancestry. So I just told you about all the insights that we can learn from these biomechanisms. And now we're looking at a complete lack of the inclusion of many different interesting communities of people. I would be in the mixed category, other. My daughter would also be in the mixed category, other. So you get the picture. So because of this, we're not learning as much as we should, and we're not accelerating the pace of those discoveries. But the far more concerning statistic is this. 72% of genetic discoveries have taken place in 3 countries on planet Earth. So if you're a student of colonialism and you understand any other extractive research-based relationship, this might look familiar to you. Africa, the continent of Africa, not to make it a monolith, it contains multitudes of people, cultures, and genetic diversity, is the most diverse place in the world genetically. Why? Because human beings have lived in Africa the longest amount of time. They've had more recombination events, etc. Classic population genetics 101. But look at their representation. How many genome sequencers exist in the continent of Africa? Base level. What is the capacity that has been built? What is the relationship? It's mostly based on extraction. Here's another example. I told you about Illumina, the largest service provider for genome sequencing. And what you'll see on the wall in their lunchroom here in La Jolla, California, is a bunch of patents on the wall. We're really good at this in America. Really, really good at it in California. And what it looks like to me is a bunch of moose heads in a hunting lodge. And you'll notice that there were approximately 45,000 patents filed worldwide that mention DNA sequencer analysis as a significant application or method. 2018 was clearly before the global pandemic, was it not? So that's accelerated, and the amount of IP that's being derived from this is flourishing, and it's not necessarily benefiting the least, the last, the looked over, and the left out. I put up the cover of one of my favorite Economist magazines. That's a British publication that I read often. And here it says that the world's most valuable resource is data. Data surpassed oil as the world's most valuable resource in 2018. When the first genome was sequenced in 2001 by our friend Craig Venter et al., the #1 companies in the world were companies like Mobil, Exxon, BP. Now it's Apple, Nvidia, Google, Tesla. What does that speak to in terms of the value and commodification of data itself? And so once you start thinking about it and understanding data as a resource and comparing it to water and comparing it to critical and rare earth minerals like we're seeing jockeying for power in Ukraine right now, oil, timber, you start to realize that there is an imbalance in the supply chain. And we're going to get to why that's important and how we're disrupting that. Tim Cook, for example, this talk that I'm giving you and many of the technologies that we rely on utilize cobalt, 75% of which is extracted from the copper belt in Congo. How many of you think that Tim Cook has ever visited the Congo to check in on his assets? He hasn't. But these images we generated using Midjourney say that he has. So look, folks, you could see, always look at the fingers, yeah? Always look at the fingers. But, so I'm just playing around, but I do think it's important to put pressure on these types of people to visit these places and become knowledgeable about the supply chain in which they're tapping into. And for those reasons, we started the Native Biodata Consortia. This is the first genome sequencing center and research institution on a reservation in the United States of America in Cheyenne River. And here are some of my partners. We're very thankful for a lot of the people that have supported our work. And this is Indigenous Futurism in action. So now I want to just turn the page a little bit and tell you about some specific projects that I've been working on in the Pacific. The first is called Paths and Timing of the Peopling of Polynesia. The second is the origins of leprosy bacterium in Oceania. And the third is about the gout pandemic in French Polynesia. And if I do my job right, you will see how all three of these projects are dialectically intertwined, okay? So let's begin. This is the Pacific Ocean, the oldest, largest, most beautiful ocean on planet Earth. And you could see that I've highlighted the Polynesian triangle. And within that triangle, we have time estimates. You know that our ancestors came out of Africa. They made their way through the Fertile Crescent. Some made a left, some made a right. Some make it through China into Taiwan. They develop double-hulled canoes and outriggers, and then they embark on what we call the original moonshot. They find places like Melanesia, Tonga, Fiji, Samoa, and then they break out into transoceanic voyaging into places like Aotearoa, New Zealand, Rapa Nui, or Easter Island, and where I'm from, the most isolated island archipelago on the planet, Hawaii. Now, we kind of take these estimates for granted. A lot of it is derived from archaeological evidence. A lot of it is, the relationships are derived from many different sources, but once you dig into the box score, it's not that clear where these estimates come from and how accurate they are and at what resolution we have begun to understand these things. There's some elegant work that's come out of many different categories, and that's why it's really important for us to find this sort of Venn diagram of agreement between different time estimates from different fields. So that could involve archaeology, carbon dating from a lot of different artifacts. That could involve applied mathematics. Brilliant work was done in the 1970s in Manchester, where they used some original simulations to identify the chance that certain island archipelagos would be found under what circumstances. Brilliant work. Then you have linguistics. Many of you work in linguistics. And we also know that there are a lot of similarities and differences. There's computational linguistics. This is a whole field that I'm not qualified to talk to you about. But I am qualified to talk to you about genetics. And it is another way in which we can understand and revisit the timing and specific routes in which our ancestors found places like Hawaii. Before we get to that, though, I wanted to talk to you about these moments of contact that occur, because they're very important. This one, for example, is a piece of art from Herb Kane of him arriving in Hawaii for the first time, and you can see like what this might look like. You know, this is how it started, right? This is how it ended, violently. It did not go well. There was miscommunications. There were questions about stealing. There were questions about all kinds of things. There are interpretations from anthropologists from around the world onto why this conflict happened and what were the circumstances and why did it end up in this situation. Because for all intents and purposes, what we know about before this happening was there was a lot of incredible collaboration going on and sharing these different ways that we know the world. There's a great poem. I won't read the whole thing, but I feel as a Hawaiian person that I have to advocate for my community and say that we're not cannibals. But I'll read it to you. It says, on cooking Captain Cook, if you ask the blonde-haired concierge at the Grand Kihei, he'll tell you that we ate him whole, strung his white meat on a stick, filed his mouth with apples, and slow roasted him over fire. The sunburned vendor selling t-shirts in Lahaina will say we ate him too, but only certain parts. The head, heart, hands wrapped in a kind of spinach and held over hot lava. The owner of the hula hula bar and grill will say we only ate him for lack of fine cuisine, rubbed his skin with sea salt, and then boiled him in coconut milk and served him on a bed of yams. My anthropology professor, long researching ancient cultures, this is for Marshall Solins, will offer explanations from his latest book. The white-skinned men seemed gods to those without metal or written words. By eating him, they meant to become him. But if you ask my tutu, my grandmother, while she waters her orchids in Protea, she will invite you in to eat. Come eat. And I think one of the most interesting things, and the distinction that I would like to let you know is, how would my people have thought that that man was a god when he couldn't even swim? Nonetheless, the reason Cook and his crew, Joseph Banks, George Vancouver, end up in the Pacific is to make scientific observations through the transit of Venus. One of the things you don't learn about in history is about his golden goose. This man's name was Tupaia. Tupaia was a master navigator said to have memorized the locations of over 1,200 different islands. And here are some of his photos that he made. He's making different watercolor paintings. He was just kind of a brilliant, well-rounded person. But I wanted to point you guys in this direction. This is a map that Cook and Tupaia created together. This is a representation of space and time and cartography, and it is lovely, and it is incredible, and it is a cross-cultural collaboration at the time. And you can imagine them pouring over whiskeys in the master's cabin, thinking about the different ways that we represent these things. And I think it's a great example of what can be done with the best tools available. And it brings us to this idea of who's in control of the narrative, who's biasing the data, and whose hypothesis are we testing? Is it even your question to ask? And I have another picture of a very famous experimental archaeologist by the name of Thor Heyerdahl. And if you're familiar with his work, he set out to prove that our ancestors came from the opposite direction, from South America, and went the other way, which we now know is not true. But kudos to him for building a boat, sailing it, and trying his hand. His book, Kon-Tiki, became very popular. You can see him here with his boat and the kind of engineering prospect that he thought would have worked. It didn't work. They had to be rescued. Now, this is in 1947. This is in stark contrast to the accomplishments of Hokulea. Has anyone ever heard of this before? Hokulea is another example of experimental archaeology, and it was because our knowledge systems were on life support, almost going extinct. It was illegal for us to speak our own language. And here you have Hokulea sailing from Honolulu to Papeete in Tahiti. I think they did it in 30 days. And what they were demonstrating is that we are the greatest seafaring community in human history. And we can rehabilitate any type of technology in this way and do it as a reflection. I mean, I can't express to you like how proud our community was, and how that looks like a great party also. But we have this saying in Hawaii, "Ika wa mamua ka wa mahope." It means I'm walking backwards into the future. It means we shouldn't repeat the mistakes we've made from the past, and we should take advice from our ancestors. And that's why it felt so good to publish this paper in Nature with a lot of my favorite colleagues, where we were looking at paths and timing of the peopling of Polynesia inferred from genomic networks. We were able to recapitulate these networks using high-resolution genome sequence data and refine our understanding and the timing of the routes that our ancestors took. We were able to not only refine our mathematical understanding, but also utilize the names of the places that our communities refer to when we're talking about. We were able to celebrate our language in Nature, which is a British publication, which I like to publish in as much as I can. But I think that it feels good, and it's important for popularizing this type of science amongst our community and vice versa in places like this. However, we don't control what Science Magazine, American publication, says about our work. So they say, no one could have predicted. DNA offers surprises of how Polynesia was settled. It's like, no, if you would have asked my grandmother, she would have told you, right? And I think even the imagery and the pictures they've chosen. So when you're upset about something, you got to write something yourself. So I got to write this piece in Scientific American about our voyaging accomplishments. And I got to connect it to a number of new ideas and debut them. And it was a lot of fun. And I also got to put our own boat, Kiakahi. This is Mauna Kea in the background. So that felt pretty good. Okay, second story. The origins of leprosy bacterium in Oceania. Another major contact moment. So I want to go through a timeline with you. First, this is data from Kamehameha Schools, which is a very popular kind of Native Hawaiian high school in collaboration with the Pew Research Center. And you can see that on the left, we have a timeline. We're starting with the arrival of Captain Cook, 1776, and then we have the number of individuals in the tens of thousands that existed in Hawaii predicted. Then we have the first known archival piece of evidence supporting that leprosy arrives in Hawaii. And then we have the establishment of Kalaupapa. Has anyone ever heard of Kalaupapa? First Kalawao, and then Kalaupapa. This is the largest case of medical segregation prior to the pandemic in American history. There was, or is, a really beautiful island called Molokai, or Molokai, depending on who you ask. And on the northernmost tip, there are the largest and highest sea cliffs in the world. And there, I'll show you a little video. If you guys ever get the chance to visit, this flight is terrible. It's terrifying because you're in those like F1 seatbelts, you know what I'm saying? And they tell you when you get on, if you weigh over 200 pounds, you gotta sit on this side of the plane. You never wanna hear that. But these are the largest, highest sea cliffs in the world. Kalaupapa ends up becoming a leper colony, an opportunity for quarantining people with leprosy prior to the development of antibiotics. Robert Louis Stevenson visited. He called it a prison fortified by nature, a pit of hell. It is both extremely beautiful and extremely controversial. And for those reasons and many others, I just described the political climate in Hawaii. I'm American, but I'm a reluctantly American, if that makes sense. I have 2 passports. And for those reasons and many others, there is a pessimism and distrust in Western science and the R word, research. And so when we're building projects that are community-based on subjects that are as sensitive as this, we have to tread lightly, and we have to operate with the highest level of respect. But leprosy, for what it's worth, even though we have antibiotics, is a neglected tropical disease for a reason. I mean, even today, there's still around 200,000 cases per year. And forgive me for the next few photos, they're not that nice. But there are many different strains of leprosy. It is a rod-shaped bacteria. You can see that there's a difference between Mycobacterium leprae and lepromatosis. And we know and have characterized the genome of this bug. We also know that it's involved in zoonotic transmission with different species like armadillos and squirrels, which is terrifying, and chimpanzees as well. Through our work, we also discovered that there are antibiotic-resistant strains that were identified in Samoa. So that's very, very terrifying. So there's a lot of value that comes from sequencing and understanding the genomes of different strains of leprosy. Here's just a look at that genome. For those of you that are not from the genomics world, this is a circular genome. This is a bacteria. This is the rod-shaped sort of representation of it. And recent work has also shown that Hansen's disease, or leprosy, could have valuable insights into how we understand cancer. So it's not just a neglected tropical disease. Researchers from the University of Edinburgh are showing that leprosy bacteria can promote host cell reprogramming, like a stem cell. Now, this is really new and nascent work, but learning more about it could be a nice entry point for scientific research. And so we worked with our oldest hospital in Hawaii, called Queen's Health Systems, founded in 1859, which isn't that old to you guys, but for us it is. And we worked in archival settings where we had FFPE, or slides from biopsies, that were stored. Now, these are ancient genomics protocols. These things have been shelved in this warehouse forever. Hawaii is not like here, it's hot. The number of variables that we're dealing with that make it difficult to extract DNA is very hard. It's basically a miracle that we were able to sequence the ancient genomes of leprosy from the Pacific. And we were able to do just that. And we were able to identify several new strains of leprosy that had never been seen before. Our quest to understand its origin is ongoing. And we published the paper in Britain. But it brings us to a new series of questions, because I showed you a picture of a population collapse. This is something we call a naive soil epidemic. That is, when communities from the New World and the Old World meet. There's introduction often of pathogens, and a lot of indigenous communities were not prepared for that. I always say from Cook to Cortez. We've observed this over and over and over again. And recently, investigators working with indigenous communities in British Columbia were able to identify bottlenecks after European contact. So I want you to think about what that means. By comparing ancient genomes of individuals of indigenous ancestry with modern communities, you can observe the dent in the immune systems of modern-day indigenous people, and you can understand how those interactions contribute to disease today. Are we following? Okay. And for those who like pictures that involve Charles Darwin, here we go. We have an original population where there's a lot of diversity. We have time. We have X pathogen. We have the creation of a bottleneck. We have a surviving population. And in this case, we have reduced HLA, human leukocyte antigen diversity, that is critical for understanding inflammatory diseases like gout. So that brings us to our last vignette here. And this project, we got to work very closely and develop the largest gout study in Polynesia's history with the French Polynesian government. And if you're like me and you love getting drained, it's awesome to work in these places. It's awesome to get to understand the health impact of strange diseases and disorders like gout. Here's a picture of some of our team and some of the different places we've got to work. It's been a true pleasure working with the Vice President of French Polynesia. They have been so accommodating for our work. It is ongoing, but I want to tell you a little bit about some of those results. So we used something called mid-pass whole genome sequencing. The reason we use mid-pass is because it's the cheapest way to get the highest level resolution data. I won't belabor it too much, but think about it as like a affordable poor man's opportunity to get nice genomes and lots of them. We also accompanied that with point-of-care technologies. So we know that de-black boxing technologies and bringing them into communities allows communities to build trust and understand what's going on. This is something called a myoton over here on the left, and it is really good for measuring things like muscle density. We also use classical old school tools like blood-based paper diagnostics and mobile ultrasound and other things. And it was very, very successful. We've published a number of papers on this project, namely in Genome Research and The Lancet, English journal. And we focused a lot on people's diet as well. No, it's not good to drink 10 beers every day, but people do it. I might do it tonight. But, and then also looking at the different types of diets. You know, one of the unfortunate circumstances of colonialism is like, when people don't have access to their land, they can't manage their land. When they can't manage their land, they can't have their traditional food sources. If they don't have access to fishing rights, they can't catch their fish. Hunting rights, they can't. You get the picture, right? And then you replace that with white rice or French baguette bread in the case of French Polynesia, and Spam, you got an issue. And what we found out was astonishing. Gout is estimated to affect 1 in 7 individuals in French Polynesia. I'm not talking about men and women. So when you dig into that, it's a strange frequency. And we had to look at the data so many different times in different ways, but what we discovered is that, yes, it is extremely high. We are experiencing a gout epidemic, and this is more work from that and looking at specifically genes involved in the MHC and HLA that are related to inflammation, regulation, and attenuation. And we've discovered allele or mutation candidates that look like they are under selection. One of the interesting things is that Tahitian communities, French Polynesian communities, it's like I showed you a bottleneck, right? And it looked like a wine bottle. But really, it's more like a telescope. It's a series of recurrent bottlenecks. You have a founder population that gets on a canoe. They end up on the canoe, and they take that canoe to another series of islands, and they are selecting specific alleles along the way. Then you have another recurrent selection event, which is our first contact meeting, which we described. And so we're just selecting, over selecting, over selecting, and we're identifying new mutations over and over and again. And one of the communities on planet Earth that is not from the Pacific that we know has had multiple founder events and selection events is in Finland. And we were able to see kind of similar results with that, which is kind of what you want to see as a population geneticist. And this is work that's forthcoming from my collaborators at University of California, Santa Cruz, Alex Ioannidis and Cole Shanks, who's a super talented graduate student. But one of the things we noticed, which was remarkable, is that if you look specifically at an allele called HLA C102, and we look at its frequency, and we look at it specifically within Polynesia versus other continental populations, we're starting to understand that when we use machine learning techniques and technologies to model the binding of every peptide segment in the proteome, we see that the highest level of affinity is for leprosy. So it's like a circular story where we've been searching for this. We've been searching for the time period it happened, and we have confirmed our hypothesis. And we will be able to do this in other populations. And it's a pretty special moment for us in terms of building our data sets in different communities. And again, this work's ongoing. We did this through a startup company that we created called Variant Bio, because you're probably asking yourself, wait a minute, Keolu, you just shit all over the commodification of DNA. And now you're telling me you're working with indigenous communities and sequencing their genomes. So what figures? And one thing you should know about this company that's unique is that we have a benefit sharing program. Has anyone ever heard of that? Okay, I'll tell you all about it. So when I go fishing with my uncles in Molokai or wherever, and we catch tons of mahi and tuna, we don't just clean it and stack our freezers. We go around in our community and we give fish to those people who are most in need. That's the same process that we use when we think about generating large-scale genome data sets from indigenous communities. So on top, we have like our traditional workflow, our data workflow, where we have collection, storage, and access. Here's a government program in America called the All of Us program. It's similar to your UK, what is it, 10K or 100K? Yeah, the biobank, the UK, 500, I'm sorry, excuse me. Okay, similar to that, 500K, excuse me. And in this case, the data just gets driven over the goal line by two pharmaceutical companies with no real regulation or governance of that mechanism. So what we have done is we've said, all right, we have indigenous or underrepresented communities. We create different sort of, you guys would call them data trusts, but these are collective interest models or individual interest models. And then we provide people with equitable benefit sharing for this. And up until this year, we had mostly worked on a really small scale where we focused primarily on infrastructure building. So let me give you some examples of benefit sharing. One is we include our community members who participated in certain aspects of our science in the author list and papers that we publish. Another is we help people get access to fresh water, like here in this project in Madagascar. We also have an affordable medicine pledge where we promise free access to the drugs that we create, which, if you're familiar with the broken healthcare system in America, you'll know that we have certain drugs that hit the market, like our new sickle cell drug, $2.1 million, Vertex. No one can afford that. Vertex, also $300,000 for a cystic fibrosis drug. These are against the common law. They should be illegal. But we not only are focused on giving people free access to the drugs, but also ensuring that our indigenous partners get a percentage of the proceeds. And up until last month, we hadn't shown our proof of concept. We had promised this, we had risked everything. We made a huge bargain. And we finally reached our first partnership with Novo Nordisk. We're giving away 4% of all of our proceeds. Now what I want you guys to think about, and we got to be on the cover of the New York Times, which was cool for my mom. But this is the first circular or benefit sharing mechanism ever in Big Pharma's history. We're very proud of this. This is a little startup out of Seattle where I went to graduate school with my partners, Kaya, and a bunch of other people. And it is disruptive, and it is revolutionary, and it is literally Robin Hood with pipettes, because we are ensuring that these communities will use that money to buy back land that was stolen from them, from their colonial overseers. From the French. I know you guys don't like the French. And that's poetic because that is the exact same land that shaped their genomes in the first place. And I think Charles Darwin would be happy about that. So in the end, I would just say that it's important to know the big currents and know the little currents. This is a famous saying that we have in Hawaii. And this is important for knowing how data flows and how it flows back to people. Okay, so before I stop, I do have a little Easter egg lecture hidden in the back here. How much time do I have? 10 minutes? Oh, perfect. Okay, we'll get to questions after. But out of all this conversation about data and machine intelligence and our applications about how these new tools are going to empower people and how getting communities to have their land back is not only good for their health and their cultural revitalization, but it's also really important for conservation. Because as I said at the top, it's really important to have indigenous people as stewards or shepherds or guardians of biocomplexity. So it's like win-win-win, right? Well, there's something going on in the background that we need to talk about. So in 2020, the world generated approximately 59 zettabytes of data. By the completion of this year, that number is expected to grow to around 175 zettabytes of data. And for those that aren't familiar or indoctrinated within the computer science community, 1 zettabyte is around 1 billion terabytes. No, I'm just kidding, but okay. But we're generating data with all these different ways. Social media, navigation, genome sequencing, IoT, even ChatGPT, obviously, and Zoom, and all of these different ways in which we're creating data. And I want to tell you something that my grandma told me. You can't have something for nothing. And if you've ever taken a physics class, you'll be familiar with the second law of thermodynamics. There's a tremendous amount of heat that we are emitting as we scale up our simultaneous addiction to both oil and data. We're running into one of the greatest problems humanity has ever faced. Part of it is thanks to NVIDIA, okay? I'm just picking on them, but we're utilizing GPUs. These are graphical processing units. And you can see that the utilization of these tools is becoming more and more easier and easier to kind of get your hands on these tools to create your own little language model, okay? How many of you are familiar with the fact that since ChatGPT debuted, Microsoft's water consumption for cooling alone in their data centers has increased 34%. That's a real relationship. You can't have something for nothing. Who's heard of the Jevons paradox or the rebound effect? Anyone? You have? You have? What is it? Precisely. Precisely. It was described in the 1860s with regards to the industrialization and refinement of coal as an energy source. So as we clean up coal, more people use it. You could apply this to pretty much anything. Jevons paradox, the rebound effect, or the backfire effect. So that's what's happening now. DeepSeek revolutionizes or upends the apple cart, literally the apple cart in this case. And we're left with technological independence and more people utilizing AI because the entry level is lower. But we've now created a multitude of people who are generating heat. You see? And so for those reasons, we've come up with different predictions. And these may or may not be right. I think they're sort of conservative. But who's familiar with Enrico Fermi, the physicist? He famously predicted the force of the Trinity tests because he had a napkin dropped out of his hand, and he calculated the force of the test within an order of magnitude based on that information. He also famously calculated the estimated number of piano tuners in Manhattan, or how many, chopsticks does it take to reach the moon? You get the picture, right? These are back of the envelope estimations of how we forecast and think about the future. So the thought experiment here is, when will data centers, server farms, and cloud computation surpass the fossil fuel industry as the #1 contributor to the climate crisis? Is that even a legitimate and logical question? How would you go about doing that? And we did some maths. And we took a look at the transportation industry alone, and we said, okay, it looks like fossil fuel use is increasing 1% every year. And it looks like our addiction to data is essentially increasing exponentially. And then these are just some estimates of when we think this will happen. But if we don't get this under control and implement new technologies and access to energy, we're in deep shit. So just keep that in the back of your mind, If you don't agree with our estimates, that's fine. We'll publish them anyway. But I want you guys to take a look at this, because this is a list of really disruptive technologies that are emerging that could help aid us as we move on to creating Earth-friendly computation tools, okay? And I've used NASA's technology readiness level scale. And the way that works is, you just say time travel, that's a 0. Genome sequencing, that's a 10. We're doing that already. And then we have technologies that kind of are occupied within the middle here. Many of them I love as a genome scientist. Look down at the bottom. DNA computing, storing data in genomes. The seeds you have in your lay, that's a data center. But there are many others. There are renewable energy sources. When you build data centers, shouldn't you be building them with renewables in mind, hyperlocal design? That's not what we're doing in America. We just said we're going to invest $500 billion in data centers in Texas. But taking this problem and looking at it through an indigenous lens is pretty valuable. Thinking about it in terms of resource flow, self-efficiency, closed-loop systems, adaptive management, prioritizing biocomputing. I mean, this large language model is about 20 trillion parameters. The LLMs that are being fired off by ChatGPT is about 2 trillion parameters. This requires 8 kilowatts per hour. I might be wrong about those specs, but my point is this is a superior device to that. And I think we need to start thinking a little more creatively. This is just our kind of news flow that's going on all over in America, the obscene energy demands of AI. AI is taking water from the desert. You get the picture. And most recently in the news, you'll notice that our new criminal in chief here, he has been centering and saying that he wants to move forward with a colonial enterprise of acquiring Greenland from Denmark. And this is a terrible, terrible, terrible idea. All kind of data would point towards the fact that they want to turn this into the world's largest server farm. And then I put this quote from Oliver Umberti, who's an awesome cartographer. And he said, Greenland has about 5,000 gigatons less ice than it had 20 years ago. Part of the problem is that once melting starts, it's hard to stop. I don't think that data centers are going to be good for Greenland. Now, if you're interested in thinking about questions in this space of Earth-friendly computation, this is sort of a brand new series of projects and grants and papers that we're working on in my group. I encourage you to join us for the next Pacific Symposium on Biocomputing in Kohala, Hawaii. And we'll be having all kinds of conversations about Earth-friendly computation and biocomputing broadly, because I think that's the future. Thank you guys so much for listening and for the opportunity, and it's lovely to be here, and I'm happy to answer everyone's questions afterwards. Thank you again.
So thank you very much indeed, Keolu. For me, you presented a really extraordinary and wonderful mixture of indigenous knowledge, modern genomics research, and some machine learning and other new technologies. But you've demonstrated how the past informs the future. And you're offering a new way of thinking and direction for genomics that I think is probably vital for the sustainability of our lives on Earth and probably the sustainability of all life on Earth. So thank you very much indeed. It's been a real pleasure.